the splicing can actually occur from one site to many different alternative splice sites. So this creates a great diversity of RNA transcripts in the cell. And then once it leaves the nucleus, it's translated into a specific protein. So how do we actually understand what proteins are being made in the cell? We have to perform RNA sequencing. So there's two major high-throughput sequencing technologies that we can use in order to understand the full array of transcripts in a cell. One, Illumina, provides a sequencing by synthesis approach, and this generates short reads of about 150 base pairs at very high quality, less than 1% error rate. Another method by PacBio provides single molecule real-time sequencing. So this produces around 1,500 base pair reads, so much longer but much lower quality, about 13% error rate. So there's trade-offs here in different sequencing methods. So once we have that sequence data, one thing we can do is align that to a reference genome. A standard alignment procedure would be to determine the alignment with the minimum edit distance, so the fewest changes that can be made, including match, mismatch, insert, and delete, in order to get this small sequence to align to the reference genome. The problem here is that this works for DNA, but not necessarily RNA, because an RNA molecule may have undergone splicing. In this case, it would, under a standard alignment, what was actually a splice from transcribing a small segment, splice over here, transcribe another segment, that would appear as a massive deletion, and it would be overly penalized as a ridiculous alignment for DNA. But that's actually totally reasonable for an RNA alignment. So a gapped alignment procedure takes splices into account and is actually able to detect novel splice sites. And one such aligner is MapSplice. So by using an aligner like that, we can actually take splices into account. So what do I mean by a splice graph? This is a data structure that we've come up with that summarizes a set of those Illumina short reads aligned using a gapped aligner to a reference genome. So it's a weighted, directed acyclic multigraph where the nodes represent genomic coordinates and edges represent connections between those coordinates. So exon edges represent those retained sequences, so the solid lines in that graph, and then dashed lines there would represent splice edges. So they would correspond to intervals that had been spliced out of those transcripts. So this graph here represents a RNA transcript that had 2 splices. And that could be much more complicated in practice with, for example, other splices extending from, say, that second node all the way down to that second to last node. So it's not necessarily a linear graph either. Then also we include SNP edges to represent significant single base variants. And I have an example of that on the next slide. SNP is a single nucleotide polymorphism, so a single alternate observed at that position in the sample relative to the reference genome. Edges are going to have an associated weight. It's a weighted graph. For exons, it's going to be the average coverage over that interval in the sample that we sequenced. And then for splices and SNPs, it's going to be the number of times that we observed that feature in a read, in one of the short reads that we aligned. So the details of how this is represented, edges are going to be directed from low to high coordinates. This makes it really easy to traverse the graph in both directions with a low coordinate that's inclusive and a high coordinate that's exclusive. That detail makes it really easy to have parallel edges for the purposes of, say, including a SNP edge in a graph like that. So therefore, that edge that goes from 963 to 964 with the 2A, that means that we saw the variant A twice at position 963 there. So that detail of how the coordinates are represented makes it really easy to include features like that that go in parallel. All edges are going to be tagged with the observed direction of transcription. DNA is double-stranded, so RNA molecules can be transcribed in either direction down the molecule, depending on the context of that particular gene. So we want to be able to include the direction there so that we know when we're later manipulating this graph what the actual context was. Additionally, exon and SNP edges are going to get tagged with a sequence. For a SNP, it's just the base in question. And for an exon edge, it's actually the reference sequence that corresponds to that genomic interval. Then, additionally, this is a multigraph, so we need to have a key for those edges in order to differentiate them in the multigraph. So for a SNP, it's the base in question. And for other edges, it's just a generic indicator of the type. So what is this data structure actually useful for? An example would be the long read alignment problem that grad students under Dr. Prins have been working on. So the splice graph can be used to correct a noisy long read, such as the ones obtained from a PacBio style sequencer. So given a splice graph built from short reads from a sample, we want to try to along a long read to that sample. So first, we can try to find a starting point by aligning small k-mers, length k sequences from that long read to exon edges. And once we've found the starting point, traverse outwards in either direction, following paths through the graph within a connected component. By doing this and performing successive alignments as we incorporate edges, and discarding edges if we exceed a local error bound in that alignment, we can then try to obtain the true path. Once we've obtained that true best path, that should correspond to the original sequence of this noisy long read, and by reading the sequence off of that sequence of edges, we can then correct it and obtain the true sequence, including variants. So what I worked on was how to construct this graph and how to include detailed variant information. So first, obtaining short read data from a Illumina sequencer, performing the gapped alignment, and then where I come in, three-phase method to construct this graph, and I'll go into detail on these on the next slides. So first, detecting splices. Each of these reads is going to have a corresponding cigar string indicating how the alignment proceeded. So it'll indicate the number of bases that it had a match, mismatch, insertion, deletion, or splice. We look through that and find the intervals that correspond to splices and extract those and insert those into the graph as new edges. Additionally, those splices may or may not be tagged already by the gapped aligner with transcription direction information. If they're tagged with that already, we can incorporate it. If not, we can inspect the sequences of the reference around the splice points, because splice junctions are going to be asymmetric with respect to the genome. So next, we have to perform a pileup of those reads, which basically means we iterate over the genomic coordinates that have some coverage, and then we will receive from this library to do the pileup a column of all of the bases at that position that had something aligned to them. We can then call variants at that position, which I'll go into more on the next slide, and we can also build exon edges into the graph. So in the example graph on the right, I have a splice extending from position 100 to 199. If we then iterate over the coordinates and see coverage from position 0 to 99, we can then start representing an edge. And then when we see the splice that happens at position 100, that'll then cause us to flush that exon to the graph and terminate that representation of the edge. Another reason that we would terminate an edge is when coverage drops below a threshold that we can configure. So if we then see no coverage from 100 to 199, and then see coverage again at position 200, we would begin a new edge. And then if that coverage dropped off at position 300, we would then insert a new edge. And by doing this process, we can account for any complicated system of splices that occurs as we have to incorporate the coverage. So the details of the variant calling procedure that would insert one of those length 1 SNP edges, like we saw earlier, is that we're given n occurrences of a base at a given position, and we're also given corresponding quality values that come from the original sequencer, representing what's the probability that this call was actually in error. So what we need to determine is an overall error probability corresponding to this particular base. And the base is a significant variant if that error probability is less than some significance threshold. So the quality values are they're defined in terms of error probabilities. So we have to do a simple conversion there in order to get that actual error probability, PI, from the quality value, QI. So the base is going to be an error as an overall variant. Each occurrence of that base is an error. So with the product rule, we just multiply those probabilities together because they're from independent reads, and that should give us the overall error probability. Then inserting that above expression and using exponent rules, we simplify that, and we get a simple expression to say, if the sum of the quality values, -1/10 of that, 10 raised to that, if that value is less than that threshold alpha, this is a significant variant, and we should include this edge in the graph. Finally, we have to propagate information about transcription direction, because initially that's only on the splice edges. So the simple approach that I've implemented already is just to take that and propagate it outward within weakly connected components in the graph. The only issue with this is if you have overlapping genes transcribed in opposite directions that have like a shared region at one end, this makes both appear to be ambiguous and transcribed in both directions. So to solve this problem, one approach that I've proposed that would be future work would be to consider each splice individually and propagate that direction information outwards until we hit another splice that tells us conflicting information. And by doing that, we would limit the scope of the ambiguous information there. So just to summarize, by constructing this graph, we can summarize all of these short reads that have been aligned and basically gain a compressed representation of all of those massive amounts of alignment information and also take into account the complexity of alternative splicing. So that's all I have. Thanks.
In this area.
What do you mean specifically?
So you put together this graph, okay, and you believe it matches up.
Mm-hmm.
But in I know in other areas that we know what the ground truth is. I don't know how you know what the ground truth is here.
Well, so we know as a level of that we know the reference sequence for the genome. We know that we're incorporating those pieces of that sequence within the graph in corresponding intervals.
So you have the sequence already existing.
Yeah. So the reference sequence itself is just a sequence that's been constructed over time by many scientists.
Okay.
So the reference
you do have ground truth. Okay.
the reference sequence is a generally accepted reference sequence. Mm-hmm.
Can you give an idea of, you said it's a compressed representation. What is the typical size of the input reads, and what is the size of your graph?
So we've worked on inputs that are anywhere from, I think, 6 to 25 gigabytes for the short read alignment files, and then ended up with results, depending on how many variants we include, depending on what that variant calling threshold is, it can be anywhere from around 200 megabytes to maybe around 400 to 600 megabytes is what I saw.
And do you also have a sense for how many nodes and edges are in the graph?
I don't have a figure in mind right now for how many are in the entire graph within a, because I mostly looked at individual connected components, which would represent a gene. But within a gene, it would usually be on the order of anywhere from 2 to maybe 5 splices, and so probably in total, usually less than 20 edges within an individual gene.